In this lecture, we will examine oxidative phosphorylation. Oxidative phosphorylation is the process in which energy generated by the electron transport chain is used to phosphorylate ADP into ATP. It is explained by the Chemiosmotic Theory. Chemiosmosis means that a chemical reaction is coupled to an osmotic gradient. As electrons pass through electron transport chain, protons are transported into the intermembrane space. As a result, an electric potential psi and a proton gradient delta pH form across the inner membrane. That's the proton motive force delta P. Protons, which are present in intermembrane space in great excess, can pass through the inner membrane and back into the matrix down their concentration gradient only through special channels. Each of those channels contains an ATP synthase. As protons pass through those channels, ATP is synthesized. There is a lot of experimental evidence to support chemiosmotic theory. So here are some observations in support of it. Actively respiring mitochondria expel protons. The pH of a weakly buffered suspension of mitochondria drops when oxygen is added. Typical pH gradient across the inner membrane is approximately 0.05 pH units. ATP synthesis stops when the inner membrane is disrupted, even though the electron transport continues. When mitochondria are placed in hypertonic solution, they swell and protons leak out. Various molecules that inhibit ATP synthesis do so by dissipating the proton gradient, which releases the energy of the gradient as heat. There are two types of ATP inhibitors. First type are uncouplers. For example, dinitrophenol. They collapse proton gradient by equalizing the proton concentrations on both sides of the membrane. Other type are ionophores. Ionophores are hydrophobic molecules that dissipate osmotic gradients by inserting themselves into a membrane and forming a channel that allows passage of ions such as protons and sodium and potassium cations. Here is again schematic representation of cellular respiration. You have seen that already in the previous presentation, in the previous lecture. So fuel molecules are oxidized in the citric acid cycle. Citric acid cycle neither generates a large amount of ATP nor includes oxygen as a reactant. Citric acid cycle is the first step in cellular respiration. It removes electrons from acetyl coenzyme A and uses those electrons to form NADH and FADH2. Each cycle generates 8 electrons, which are utilized to reduce oxygen molecule and generate proton gradient in the second step, in the oxidative phosphorylation step. And that's where most of the ATP is synthesized. The enzyme ATP synthase is embedded in the inner mitochondrial membrane on its inner surface. It is composed of two major components, F1 and F0. F1 unit has 3 nucleotide binding sites and is composed of 5 different types of subunits. They are in the ratio of alpha 3 and beta 3. Alpha and beta subunits form dimers. So one alpha and one beta unit together form one dimer. And that means since there are 3 alpha and 3 beta units, there are a total of 3 alpha beta dimers. So that's a major part of F1 component. Then there is also gamma component, which is part of the rotor, as you will see in a minute. Then delta component, and finally eta component. So this is F1 part of ATP synthase. F0 unit is a transmembrane channel for proteins. So proteins pass through it and is composed of 3 different types of units. in the ratio of A, B2, C12. So there is one A sub A unit, 2 B units, and 12 C units. Translocation of 3 protons through the ATP synthase is required for synthesis of each ATP molecule. An additional proton is required to transport ATP and hydroxide out of the matrix in exchange for ADP and inorganic phosphate. So that means a total of 4 protons are required for synthesis of one ATP molecule. The rotor is composed of units eta, gamma, and C12, while stator is composed of A, B2, delta, A3, and BT units. So here we have still highlighted units, highlighted F1 in yellow and highlighted F0 in blue. So now you can see ATP synthase better, and you can see rotor and stator. So rotor is basically in green. You can see at the bottom 12 C units that form part of the rotor, and also gamma, which is basically a shaft that connects rotor to alpha beta dimers, and small eta unit. So C, gamma, and eta are rotor, rest is the stator. Proton motive force induces a 3-step rotation of 120 degrees. 3 steps, that means that once 3 protons pass through proton tunnels, actually there are, or proton channels, there are 2 proton half channels, you can see them in red. Once proton enters bottom half channel from the intermembrane space, it binds to aspartate that is part of unit C. Once 3 protons bind, so sequentially one after another, that results in 3-step rotation, and that's a total of 120 degrees rotation. As protons flow through F0, rotation is transferred to the gamma subunit, and that projects into alpha beta dimers or core of F1 component. Rotation of gamma subunits puts these alpha beta subunits into 3 possible positions. And binding activity of the catalytic beta subunit changes with alternating position of gamma subunit. So we can identify 3 conformations. Conformation A is called open conformation. It is inactive and weakly binds ADP and inorganic phosphate. So that's first step in the synthesis of ATP. Conformation B is called tight conformation, or T, and is active and brings the substrates together to form ATP. Finally, conformation C is called loose conformation. It is inactive, non-binding, and releases ATP from the active site. And so this final step makes the overall process favorable. Direction of the proton flow determines the direction of the rotor rotation and direction of the reaction, which leads towards synthesis of ATP. There are some pretty good animations that illustrate oxidative phosphorylation and ATP synthesis, and so I would recommend that you see them. And there are additional animations and additional information that is available at NDSU website. I think that's North Dakota State University. So very good animations, and I would recommend that you see them. Control of oxidative phosphorylation allows the cell to produce only the amount of ATP immediately required to sustain current activities. Under normal circumstances, electron transport and ATP synthesis are tightly coupled. The PO ratio, or phosphorus-oxygen ratio, reflects degree of coupling between electron transport and ATP synthesis. The measured maximum rate for the oxidation of NADH is 2.5 and FADH2 is 1.5. So PO ratio is number of moles of phosphate that is consumed divided by number of moles of oxygen atoms reduced to water, or phosphate over oxygen. Oxidative phosphorylation occurs only in the presence of the starting materials, ADP and phosphate. ATP synthase is controlled by the product-reactant ratio. It is inhibited by the high concentration of ATP, so product inhibition, and activated by high concentrations of ADP and phosphate, feed-forward stimulation. Concentrations of ATP, ADP, and phosphate within mitochondria are controlled by the two transport proteins, the ADP-ATP translocator and the phosphate carrier. And finally, we are going to look at the two transporters. The first one is ADP-ATP translocator. ADP-ATP translocator is a dimeric protein. It carries out stoichiometric 1-for-1 exchange of mitochondrial ATP for ADP produced in the cytoplasm. Inner mitochondrial membrane has a negative potential on the inside and a positive potential on the outside. As ATP has one more negative charge than ADP, an outward transport of ATP and inward transport of ADP are thermodynamically spontaneous reactions. It means that this process and this transporter do not require any energy. The process is spontaneous. Second transporter is the phosphate translocase. The phosphate translocase mediates transport of dihydrogen phosphate anion along with proton. It is a diphosphate anion proton symporter. A symporter is a transporter that transports both species in the same direction. The inward transport of 4 protons is required for synthesis of one ATP molecule. 3 protons participate in synthesis of ATP, and 1 additional proton is required for transport of phosphate. So this completes our lecture on oxidative phosphorylation.